tab, you press it on ink pad. Okay, like this. Okay, and then after that, you take a white paper and then you press the tab with ink on it so that it leaves the pattern of the fingerprint. Okay, and then after that, you're supposed to take a hand lens and study the pattern. And in the case of my pattern, I can compare this with what I have there on the board, and I can see that my type is loop. Okay, so that is the third example of discontinuous variation. That gene mutation is a change in the sequence of nucleotides in a segment of DNA. Change in the sequence of nucleotides in a segment of DNA. Remember, we know what is a DNA is. We have already discussed it. So you know that a DNA is a molecule which carries which is composed of the various genes that determine like that. You should never ever use the same color as the same letter as in complete dominance. You use different colors to represent the two alleles. Why? Because the two alleles behave as if they are independent of each other. Okay, so you have to use different colors. So in the case of the red-flowered Mirabilis jarapa, the allele for red flower is R, while the allele for white flower is represented by letter W, and both of them are written in capital letters. So a good example is crossing between a white flowered Mirabilis jarapa and a red flowers Mirabilis jarapa. So how do you work out such a problem? So I said for the white, for allele for the white color, allele for the red color, you're supposed to use different letters, R and W, so that structure formed next to the template. After unwinding and unzipping, nucleotides will come and align themselves along the DNA, the parent DNA strands. They will come and align themselves. And then they align themselves according to the base sequence on the DNA strand. Remember, a nucleotide has a base attached to it. And that base will come and look for the complementary base along the strand. And then they will come and align themselves along the strand until a new strand is formed, as shown there on the board. You can see that a new complementary strand has been formed on DNA strand 1, and then we have DNA strand 2. We have another new complementary strand being formed there. And then after the new strands have been formed, then they coil around each other. That's the new complementary strand and the original strand coil around each other. And then you end up having now two DNA structures which are the same in structure. They are the same structure, same in length, same in everything. Okay, so and that is what we call a complementary. That's what we call a new. DNA replication, because new DNA structures have been formed which are similar to each other. Okay, every set of 3 bases along the messenger allele is responsible for bringing a particular type of amino acid for